Imagine trying to patch a really delicate, like hyperelastic silk hammock with a rigid steel chain link fence.
Oh, wow. Yeah, that's quite the image.
Right. Because, I mean, the chain link fence itself might be incredibly strong. It might hold up under pressure, but every single time someone gets into that hammock or every time the wind blows and it tries to sway, that rigid steel is going to just violently shred the delicate silk around.
It's going to destroy the very thing it's trying to support.
Exactly. And for decades, that is sort of exactly the kind of mechanics the medical field has been applying to women's bodies. So welcome to today's deep dive. We are exploring a really massive fundamental shift happening right now in women's health.
It really is a huge paradigm shift.
It is. And the mission for you listening today is to understand this complex but honestly thrilling transition from a mechanical hardware fix to a biological software update. We're asking one really specific question. Can exosome-mediated tissue repair improve structural hammock support under the bladder neck in mild stress urinary incontinence?
And, it is a question that requires us to completely rethink how the human body heals itself.
Yeah.
To tackle this, we are pulling from a pretty diverse stack of sources today. We're looking at surgical technique overviews of subfacial hammocks, cutting-edge preclinical studies from places like the Mayo Clinic and, regenerative medicine journals that are mapping out these cellular pathways.
Plus the legal side, right?
Right, yes, even the strict regulatory compliance documents from Exavera Sciences. We're basically going to look at how we went from installing rigid plastics in the body to, well, programming the body's own cells to rebuild themselves from the inside out.
Which is just wild to think about.
Yeah.
But to truly appreciate that software upgrade, we have to look at how devastating the hardware failures have been. And to do that, we really need to talk about stress urinary incontinence, or SUI, because the numbers here are just, they're staggering.
They are. The prevalence literally cannot be overstated.
Like, this affects up to 50% of adult women, half the adult female population.
Yeah, and we see the incidence peaking at two main points in a woman's life. So first around age 46, and then a second peak later, around age 70.
So what's actually happening mechanically?
Well, functionally speaking, SUI occurs when the physical pressure inside the bladder exceeds the closing pressure of the urethra.
Okay.
For someone living with this, it means that during physical exertion, or even just like everyday actions like coughing, laughing, picking up a grocery bag, or sneezing, there is involuntary urinary leakage.
So if the bladder pressure is overpowering the urethra, what exactly is failing? I mean, what is supposed to be keeping that urethra closed against the pressure in the first place?
Right, so continence relies really heavily on a structure we often refer to as the hammock.
The silk hammock from the analogy.
Exactly. It's located directly under the bladder neck.
Right.
But it's not just a simple sling, It is a highly complex neuromuscular unit. The foundation is the pubococcygeus muscle. But weaving through and around that muscle is this highly specific connective tissue matrix,
Made of what?
primarily of collagen and elastin.
Okay, I want to break down those materials for a second. If the pubococcygeus is the actual muscle doing the contracting, what role do the collagen and elastin play in that hammock? Are they just like the biological scaffolding?
They are, but they're dynamic scaffolding. So
Okay.
collagen provides the tensile strength, which is the ability to resist being pulled apart.
Right.
And elastin provides the stretch and the recoil, the bounce back. Together, they create what biomechanical engineers call an anisotropic hyperelastic tissue.
Anisotropic. What does that mean in this context?
Anisotropic basically just means it behaves differently depending on the direction of the force applied to it. Think of pulling on a knitted sweater.
Oh, okay.
If you pull it vertically, it behaves one way, but if you stretch it diagonally, it behaves completely differently. The pelvic floor has to be able to stretch and absorb pressure from multiple angles without snapping.
But, I mean, it goes through intense, extreme trauma.
Immense trauma, like during human childbirth, those pelvic floor muscles and the surrounding matrix, they have to elongate by up to 300% of their resting length.
300%. That's unbelievable.
It is. And that degree of mechanical strain frequently exceeds the tissue's biological capacity to adapt. So the result is micro tears, severe muscle avulsions, and a fundamental breaking apart of that delicate elastin and collagen matrix.
And then you factor in that second peak you mentioned earlier, around age 70, because you have the natural aging process plus the severe loss of estrogen during menopause, which, from what I understand, actively degrades connective tissue.
It does, yes. Estrogen is crucial for maintaining that tissue.
So you have physical tearing compounded by hormonal starvation, and that hammock literally just begins to sag.
Which brings us to how medicine traditionally responded to this. For a really long time, the gold standard for a sagging pelvic hammock was this relatively quick 15-minute reconstructive surgery using synthetic non-absorbable polypropylene meshes. Right, PP mesh. A surgeon makes a vaginal incision and literally physically inserts a strip of this plastic tape. The goal is to mechanically prop up the subfacial hammock and, support the urethra.
See, listening to this, it honestly sounds exactly like patching that silk hammock with a chain link fence. You are taking a hyperelastic, multidirectional living tissue, and you are just shoving a rigid piece of synthetic plastic into it. What exactly happens to the surrounding tissue when the body realizes this plastic is in there?
The biological interaction is, well, it's often disastrous. The body immediately recognizes this rigid polypropylene as a foreign invader.
Of course it does.
And this triggers a massive localized immune response called a foreign body reaction. The body realizes it cannot break down the plastic, so it basically tries to wall it off.
How does it do that?
Specialized immune cells called macrophages swarm the area, and they just begin depositing thick, rigid scar tissue all around the mesh.
So you get this chronic localized inflammation.
Severe inflammation and severe fibrosis. And because the mesh is stiff and the surrounding pelvic tissue desperately needs to be flexible to actually function, the presence of the mesh and all that resulting scar tissue can actually cause the surrounding tissue to contract by up to 60%.
Wait, 60%?
Yes. The tissue pulls incredibly tight around the plastic.
That sounds excruciating.
It leads to a horrific cascade of complications. I mean, patients experience intractable chronic pain. The tissue contraction and constant friction cause the mesh to physically erode into the bladder or the vaginal wall.
Oh my God.
We are talking about a remarkably high complication rate of 10 to 30% within the first 5 years of implantation.
I mean, 10 to 30% is a massive failure rate for a surgical intervention meant to improve quality of life.
Yeah.
If we knew the pelvic tissue is hyperelastic, meaning it inherently needs to stretch and bounce back, why did we ever think rigid synthetic plastics were the right long-term solution?
Well, the medical field was basically looking at a mechanical symptom, a sagging structure, and applying a purely mechanical fix. The tissue was weak, so we gave it a crutch.
Just a structural problem.
Exactly. But we completely ignored the underlying biology of how that tissue survives and functions. And the complications eventually became so undeniable that the FDA had to issue public health notifications warning against the use of vaginal mesh for pelvic organ prolapse.
And didn't some countries just ban it completely?
Yes. In countries like Australia, New Zealand, and the UK, transvaginal polypropylene mesh has been outright banned.
Which leaves millions of women with this bimodal peak of SUI, and suddenly the primary tool in the surgical toolbox is just gone.
So medicine had to pivot. If the hardware is causing the body to attack itself, we have to look at the biology, right? We have to figure out how to heal the tissue itself.
And the initial pivot in regenerative medicine was toward live stem cells, specifically mesenchymal stem cells, or MSCs.
And those come from where? Usually derived from a patient's own fat, bone marrow, or endometrial tissue. The theoretical model was really straightforward. Inject these live stem cells directly into the damaged pelvic floor, where they would divide, differentiate, and just become brand new muscle and connective tissue.
But stem cells carry a massive amount of clinical baggage. It's not as simple as injecting them and walking away.
And far from it. Live cell therapies present immense clinical hurdles. First, there is the risk of the cells dividing uncontrollably and forming tumor.
Right, which is obviously terrifying.
Then there is the risk of immune rejection if the cells aren't perfectly matched. But practically speaking, there's the sheer logistical nightmare of keeping live cells viable.
Because they're alive, you have to treat them like living things.
Exactly. You have to culture them, keep them alive during transport, and inject them quickly. And even if you do all of that perfectly, a massive percentage of injected stem cells simply die shortly after being introduced to the harsh, inflamed environment of a damaged pelvic floor.
Now, this is the part of the research that absolutely blew my mind. Because researchers made a breakthrough discovery about those stem cells. They realized that the stem cells weren't actually sticking around and transforming into new muscle or collagen themselves. They were doing something else entirely.
Yes, the paradigm shifted completely away from the cells themselves and toward the secretome.
The secretome.
Right. The secretome is the collection of substances that a cell secretes into its environment. Researchers realized that the primary therapeutic benefit of stem cells came from their paracrine action.
Paracrine.
Paracrine just refers to cells communicating with neighboring cells. The stem cells were basically acting as medicinal signaling centers. They were releasing these tiny vesicles into the surrounding fluid to communicate with the host's existing damaged cells.
And those tiny messenger vesicles are the exosomes.
They are. Exosomes are extracellular vesicles, meaning they exist outside the cell, and they are unimaginably small, ranging from just 30 to 150 nanometers in diameter.
That's hard to even picture.
To put that in perspective, a single human hair is about 80,000 nanometers thick. Exosomes are secreted by nearly all cell types, but crucially, they are completely cell-free. They do not contain a nucleus. They are not alive.
Okay, let me make sure I'm wrapping my head around this. If the damaged pelvic floor is a construction site, the stem cells were initially thought of as the construction workers we were bringing in to rebuild the site.
Right.
But what we actually discovered is that exosomes are the blueprints and the text messages those workers use to coordinate the build. We realized we don't need to transport live, fragile workers to the site with all the logistical nightmares that entails. We just need to deliver the blueprints directly to the local existing cells so they know how to rebuild themselves.
That framing hits the nail on the head perfectly. And the advantages of delivering just the blueprints are profound. Because exosomes are completely acellular, they bypass the risk of immune rejection.
Because there's no cell for the immune system to recognize as foreign.
Exactly. The body doesn't see them as foreign invaders in the same way it sees foreign cells or plastic mesh. They don't have the machinery to divide, so they don't form tumors. And from a practical clinical standpoint, because they aren't alive, they can be freeze-dried.
Lyophilized.
Right. They can be lyophilized, put in a little vial, and just stored on a shelf for months. You don't need complex million-dollar live cell culture facilities at the local hospital.
Okay, so we have these microscopic blueprints. We know they are stable and safe. But what does this actually mean for SUI? Like, if exosomes are delivering instructions to the damaged pelvic floor, how are they entering the cells and what exact instructions are they delivering?
To understand the mechanism, we have to look really closely at the preclinical models. These are studies done on rats and pigs that have simulated birth trauma or urethral injuries.
Right.
When an exosome reaches a target cell, its outer lipid membrane actually fuses with the membrane of the damaged cell. It acts like a tiny USB drive plugging into a computer, dropping off a highly specific payload of proteins, lipids, and a very important molecule called microRNA.
Okay, let's start with the muscle, the pubococcygeus muscle. What does that microRNA tell the muscle to do?
The exosomes, particularly those derived from urine-derived stem cells or adipose tissue, actively promote myogenesis, which is the formation of new muscle tissue. The microRNA they drop off targets dormant cells called satellite cells.
Satellite cells?
Yeah, satellite cells are basically the muscle's own local reserve team, just waiting to be activated.
So the exosome plugs in, drops the microRNA, and wakes them up.
It does this by activating specific cellular pathways, primarily the ERK and NFP pathways. Think of these pathways as molecular highways inside the cell.
Okay, I'm with you.
The microRNA acts as a signal that travels down these highways, straight to the cell's command center, instructing it to wake up, proliferate, and differentiate into new, healthy muscle fibers. In the animal models, we literally see them rebuilding the atrophied sphincter muscle.
Okay, I hear you saying they instruct cells to, wake up and multiply, but I really have to push back here. Isn't cell proliferation exactly what cancer is? I mean, if we're delivering blueprints that tell cells to multiply rapidly, how do we know?
Exactly. Once the repair pathways like the ERK pathway have done their job and the microRNA degrades, the signaling simply stops. It is a controlled burst of regenerative signaling, not a permanent genetic mutation.
Oh, that makes a lot of sense. So that handles the muscle repair. Yeah. But what about the connective tissue? The actual hammock part made of collagen and elastin. If that matrix is shredded, how do the blueprints fix it?
The mechanism here is remarkably elegant. The exosomes fuse with the host's fibroblasts. And fibroblasts are the cells responsible for building connective tissue.
Okay.
Once inside, the exosome cargo upregulates specific genes, most notably the COL1A1 gene. And when this gene is turned on, the fibroblast kicks into overdrive, dramatically increasing the synthesis of type 1 collagen and elastin.
They are literally printing new silk for the hammock.
They are, but building new scaffolding isn't enough if the environment is still toxic. Remember, a damaged pelvic floor is usually stuck in a state of chronic degradation.
Right, because of the trauma.
So the exosomes also carry signals that do the exact opposite. They downregulate matrix metalloproteinases, or MMPs.
And MMPs are the enzymes responsible for degrading collagen? They're basically the demolition crew.
Yes. In a damaged, inflamed tissue, the MMP demolition crew is just running rampant, breaking down collagen way faster than the body can build it.
Oh, wow.
By downregulating the MMPs and simultaneously upregulating that CO1A1 gene, the exosomes effectively stop the demolition and restart the construction, actually reversing the structural breakdown of the hammock.
And what about the inflammation? We talked earlier about how the plastic mesh caused a massive foreign body reaction.
Yeah.
Does the tissue just stay inflamed with this?
The exosomes handle the inflammation by directly reprogramming the immune system. They promote something called macrophage polarization. We mentioned macrophages earlier. They are the immune cells that swarm an injury.
Right, building the scar tissue.
When they are in what we call an M1 state, they are pro-inflammatory. They cause swelling, collateral damage, and scarring.
They're the attack dogs.
Exactly. But there is an alternative state, the M2 state. M2 macrophages are anti-inflammatory and regenerative. They clean up the debris and calm the area down.
And the exosomes can switch that.
They carry signals that force the local immune system to switch from the M1 attack state to the M2 regenerative state. This shuts down the chronic inflammation and entirely prevents that terrible scarring and fibrosis we used to see with the synthetic mesh implants.
The coordination of all these different signals is just astounding. You have muscle regeneration, collagen synthesis, enzyme suppression, and immune system reprogramming all happening at the exact same time.
Yeah.
But does it actually work? Does the hammock actually function again, or does it just look pretty under a microscope?
Oh, it functions. The histological evidence, the tissue biopsies, shows new blood vessel formation, thicker muscle fibers, and a much denser collagen matrix. But the functional recovery is definitely the most important part.
Right, because that's what matters to the patient.
Exactly. When researchers perform urodynamic testing on these animal models, they measure something called leak point pressure, or LPP. This is the amount of abdominal pressure required to force urine out.
Okay.
In the exosome-treated models, the LPP significantly improves. The physical hammock is functionally restored. It can actually withstand the pressure of a cough or a sneeze without leaking.
For those of you listening, really think about what this means. Instead of going into an operating room to have a permanent piece of plastic surgically bolted into your pelvis, we are looking at a biological process that teaches your body to heal itself. But I mean, that brings up a massive practical engineering hurdle.
The delivery problem.
The delivery problem. Exosomes are microscopic. If you inject a liquid solution of these nano-sized vesicles into the pelvic floor, what stops them from just, washing away into the bloodstream or being flushed out by tissue fluid? How do you keep these blueprints exactly where they need to be, right under the bladder neck, long enough for the local cells to actually read them?
That is exactly the engineering puzzle researchers at the Mayo Clinic had to solve. They developed a purified exosome product, or PP, derived from human platelets, but they knew a simple liquid injection just wouldn't work.
Because it would just dissipate.
Right. So to solve the delivery issue, they mixed the exosomes into a specialized biological carrier, a hydrogel made of collagen-1 and fibrin.
Oh, so it's not a free-flowing liquid anymore.
Not at all. It acts as a scaffold. When this mixture is injected, the hydrogel reacts to human body temperature and provides a localized, highly sustained release of the exosomes.
That's brilliant.
If you look at it under a scanning electron microscope, the exosomes trapped in the hydrogel actually look like a bead on a string pattern. And in their porcine models, using pigs that mimic human stress urinary incontinence, this biopotentiated hydrogel successfully remained exactly where it was injected.
It didn't wash away.
It didn't. And it successfully induced the functional restoration of the external urethral sphincter.
It is mind-blowing that we can literally rebuild a sphincter in a Mayo Clinic pig study. But okay, that leads to a really harsh reality check. Because if a woman is listening to this deep dive right now, struggling with SUI, and she wants this treatment, what is legally stopping her doctor from just ordering a vial of exosomes tomorrow and injecting it?
Ah, the regulatory landscape.
Yeah, because if you look around the aesthetic gynecology space right now, you see an explosion of exosome products. There are protocols, like the exofemale protocol, using adipose-derived exosomes post-laser or microneedling for vaginal rejuvenation, hydration, and tissue elasticity.
The aesthetic space has definitely adopted exosomes very rapidly, but applying an exosome serum topically for aesthetic hydration or post-laser recovery is in a vastly different legal and regulatory universe than injecting an exosome hydrogel deep into the pelvic floor to cure a complex medical disease like stress urinary incontinence.
And this is where we have to look closely at our regulatory sources, specifically the compliance breakdown from Exavera Sciences regarding where exosomes actually sit with the FDA in 2026. Because the rules governing regenerative medicine are, well, they're incredibly strict.
They are, and the distinction really comes down to two FDA regulatory pathways.
Mhm.
The 361 pathway and the 351 pathway.
Break those down for us.
A 361 HCTP product is a human cell or tissue product that reaches the clinic relatively easily, simply through registration and listing with the FDA. It is kind of a fast track.
Okay.
But a 351 biologic is classified essentially as a drug. It requires an incredibly rigorous pre-market review, multi-phase human clinical trials, and an approved biologics license application or an IND for investigational use.
And I imagine every regenerative clinic in the country wants to argue that their exosome vials are just simple 361 products so they can sell them immediately.
They certainly attempt to make that argument, but the regulatory documents make it crystal clear that exosomes fail the criteria for the 361 fast-track pathway on two major undeniable fronts.
Right.
First, to be a 361 product, the material must be minimally manipulated. It cannot be significantly altered from its natural state. But the entire process of isolating, culturing, and concentrating exosomes fundamentally alters the biological characteristics of the starting material.
Because you are actively extracting and concentrating the active blueprints, that is way more than minimal manipulation.
Exactly. And the second failure is even more straightforward. A 361 product is legally defined as containing human cells or tissues.
Oh, right.
But as we established at the very beginning of this discussion, exosomes are explicitly acellular. They are not cells.
So they don't even qualify under the definition.
Right. Because they fail both of these core criteria, any exosome product intended to be injected to treat a disease or condition in humans is strictly regulated by the FDA as a Section 351 biologic.
So if someone sees a clinic right now in 2026 advertising a direct exosome injection as an outright approved cure for their SUI, are those clinics essentially violating FDA regulations?
If they are marketing it as an approved therapeutic cure for SUI outside of an authorized FDA-monitored clinical trial, they are entirely jumping the gun. The FDA has stated unequivocally that claims suggesting exosome therapies for diseases fall outside of biologic provisions are simply untrue.
Wow.
The science we discussed today is incredibly promising, and the preclinical animal data is robust, but the regulatory pathway for a 351 biologic takes time. And it takes time for a reason: to ensure absolute safety, dosing accuracy, and long-term efficacy in human populations before it becomes widely available.
What a profound journey this deep dive has been. I mean, we started by looking at the somewhat medieval mechanics of the past, the idea that you could fix a hyperelastic living pelvic hammock by surgically shoving a rigid synthetic plastic mesh into it.
Right, a mechanical fix for a biological problem.
Exactly. And we saw how that hardware approach led to localized immune warfare, severe scarring, and just terrible complication rates for women. But then we looked at the biological future. We explored how science shifted to a software approach.
We saw the field move past the complications and tumor risks of live stem cells and instead isolate the secretome, the tiny cell-free exosomes that act as biological blueprints.
We unpacked the incredible cellular mechanisms, how those tiny blueprints use cellular highways like the ERK pathway to wake up dormant satellite cells to build new muscle, how they reprogram fibroblasts to print new collagen while simultaneously stopping the enzymes that tear it down.
And how they flip the immune system from attack mode to healing mode.
Yeah. And finally, how innovations like hydrogels can keep these microscopic messengers locked in place to do their work, even as the FDA carefully manages their safe transition into mainstream clinical approval.
It really represents a fundamental philosophical shift in how medicine views healing. We are no longer trying to act as carpenters, mechanically bolting tissue together. We are learning to act as programmers, providing the body with the specific code it needs to orchestrate its own repair.
Which brings us to our final thought. As we wrap up this deep dive, what is the one provocative idea or question you want to leave the listener with today?
Well, if we connect all of the cellular programming to the bigger picture, think about where this technology goes next. If we can isolate specific exosomes that carry the exact microRNA instructions to upregulate collagen and repair the pubococcygeus muscle, could we one day map a woman's specific genetic tissue deficiency beforehand?
Oh, wow.
Essentially, instead of waiting for the pelvic hammock to physically tear and cause stress urinary incontinence, could we deliver a custom-tailored exosome hydrogel to program her body to continuously perform preventative maintenance on its own pelvic floor before the damage ever occurs?
Treating the cellular breakdown before the physical hardware ever shows a crack, programming the body to constantly reinforce its own hammock. I mean, that completely redefines the landscape of preventative medicine. To our listener, thank you for joining us on this deep dive. Keep questioning, keep learning, and we'll catch you next time.